Over 70% of Aotearoa New Zealand's freshwater fish species are at risk or in decline. In our lakes, native fish are facing a series of challenges, from the introduction of invasive species to the intensification of agriculture. Surveying fish populations in our lakes is resource heavy, so we often end up making a lot of assumptions about what's really causing native fish to decline. But despite what might seem like a bleak outlook for our native fish species, this in fact is a really exciting time. Using paleolimnology and recent advances in environmental DNA in my PhD, I explored what is causing the decline of our native fish communities and what action we can take. In paleolimnology, we study the sediment layers that accumulate over time at the bottom of a lake. These layers are like an environmental storybook, capturing the history of the lake. Every living organism leaves traces of its DNA in the environment. By extracting this DNA from environmental samples such as lake sediments, we can identify which species are or were present in these environments. So I looked at fish eDNA in lake sediment to understand how native fish communities have changed over the last 1,000 years. I linked these changes to various environmental pressures to pinpoint which ones were driving declines or impacting our native fish populations. With this new knowledge, we can more effectively manage the pressures that are directly impacting our native fish, improving both their protection and management, along with the health of the wider ecosystem. As we develop this technology, we're expanding this approach to study entire ecosystems and overall lake biodiversity as part of the Our Lakes, Our Future program. While there are big challenges ahead for our freshwater ecosystems, our hope is that tools like eDNA monitoring will help us to better understand the issues and find new solutions that will help us to restore the health of our lakes and protect our native fish now and into the future.